Hi, my name is Franklin Alongi. I am a undergraduate student at Montana State University in Bozeman, Montana, and I'm also a undergraduate researcher in the lab of Dr. Daniel Ulrich. So my project that I'm going to be talking about right now is drought and beetle-induced monoterpene defenses in pinyon pine seedlings, which is kind of a mouthful. So first, I'm going to talk about the problem. So what the issue that pinyon pine seedlings or pinyon pine as a species has been facing in recent years. Then I'll go over how that drought that it's been experiencing has influenced our study design and our 3 different treatments that we impose on our seedlings. Talk about the results that we have so far from this data. It hasn't been fully analyzed, but we have a good starting basis, and which is allowing us to form some future hypotheses, which will be affecting our going forward and our future work. So here's the issue. In 2002, in the southwest of the United States, there was a massive drought event, which lasted, I believe, for an entire season or multiple years, which led to an estimated mortality of right around 350 million pinyon pine trees. Now, this, of course, is just an estimate, but the best guess is that this occurred over about 1.2 million hectares, or about the size of Hawaii. Now, what that actually looks like is this. So this map in green shows Pinus edulis, which is pinyon pine, just the geographic range of where it normally can grow, not necessarily where it is. The red here is data that was taken from aerial surveys done by the University of Northern Arizona, which every year following 2002, I believe through 2009, tracked just where the mortality, basically looking on the ground and seeing where are there just splotches of brown trees that died from drought, where did this occur? And they tracked it every year to see how it spread, right? So if the drought occurred in 2002, that means that in the subsequent years, the outcome from that, it'll be felt for a while. So that's why they do these continuous surveys. As you can see, it takes up both the range and the mortality occurred over a very, very large area. So mainly across 4 different southwestern states, but also the range extends into California and Texas, and most likely also Nevada and Mexico. But this mortality was more or less universal across this range and affected quite a lot of the species. But the issue is not only the drought. So drought is a abiotic factor, but there's almost always interactions with biotic factors as well. And that's where bark beetles come in, Ips confusus. So here in Montana, bark beetles are very prevalent. They lead to the mortality of a bunch of our high elevation pine species. And this is also the case in the Southwest as well. But bark beetles don't always have that same detrimental role. So in the Southwest, they're actually considered secondary pests. And what that means is that they mainly target and affect weakened trees, or trees that are already experiencing some sort of physiological stress. And what that does as an ecological role is kind of beneficial. It acts as a thinning role. So we think of bark beetles as being this evil, killing our forests kind of thing, but in their normal sort of ecosystem role, they're actually helping the environment stay healthy. So by targeting these weakened trees, these stressed trees, or these almost dying trees, they're really thinning out the ecosystem and allowing for future growth. But this can be an issue when you have a massive drought event, because then you go from there being a relatively stable ecosystem to having very, very large amounts of stressed and droughted trees. This creates a safe haven for these bark beetles to really propagate and move beyond the secondary pest role into more of a primary role, where they are attacking trees that normally are healthy and affecting the ecosystem at a much larger scale. Now, the way that they do this is primarily through the production of aggregate pheromones. Aggregate pheromones are produced when a beetle detects a tree that is weakened. They produce these pheromones, which then attract other beetles. So it can go from a single beetle finding a tree or a stand of trees that are drought stressed, and then being able to instigate this massive community-wide, population-wide attack. What they do is they bore into the phloem, or the nutrient transport tissue of the actual tree, and can effectively girdle the tree, killing its ability to transport resources throughout the tree. Now, of course, drought is bad because it leads to a haven for these beetles, but also recent changes in climate have created kind of a similar effect for these beetles. So not only are the growing seasons longer in the year, which allows for more successful or more repetitive beetle generations, but they're also just warmer climates, which allow these beetles to have multiple more generations than normal in the typical growing year, and really, really increase these populations. So our study design, we are looking at sort of these interacting effects between drought, stress, physiological stress, beetle pheromones, and how these work, and the defense response. So that's what we're really trying to get at with these treatments. So our first treatment that we did was just whether the tree received water or was droughted, which is pretty straightforward. Then secondary, we're looking at methyl jasmonate. Methyl jasmonate is a compound which plants typically produce upon contact with a beetle that's attacking it. So like I said before, the beetle will bore into the bole of the tree. And the tree knows that, it detects that, right? It creates a defensive response. And one of the first things it does is produce methyl jasmonate, which then can turn on other systematic defenses that the tree has. Now, in some species, trees have actually, or species have been able to directly detect methyl jasmonate from a neighboring tree. So imagine there's two plants next to each other. One is attacked. It produces methyl jasmonate for its own defenses, but the neighboring tree can detect it. So we're curious if this is a similar thing that was observed in pinyon pine. Beetle pheromone, similar to methyl jasmonate, has the ability to be detected in some species. This has not been detected in pinyon pine necessarily. So we were curious if we expose these trees to the pheromone that the beetles produce, if they are able to mount some sort of defensive response from that detection. And in total, we had 96 seedlings equally split up across these different treatments. But what's important is what even are monoterpenes and VOCs, which is what we're measuring here. So VOCs just means volatile organic compounds, and that can include monoterpenes, sesquiterpenes, and a variety of other things. When you break open a tree or a needle, you crush it up and it smells good, you're really smelling those VOCs. So to put it in layman's terms, they're just chemical compounds that are produced by the plants. They are primarily used in defense, which is why they're found in resin. So resin, when you break a branch off and that tree gets a little sticky, it's producing this defensive response to seal off that wound. But additionally, if a bark beetle were to bore into a tree, it will produce that resin to physically push that beetle out of the tree. It's a very effective mechanism. But not only is it a physical mechanism, but those chemical compounds, those monoterpenes and VOCs that are within that resin, are actually extremely detrimental to the bark beetles. They're found to be very effective at chemically killing while also physically pushing those beetles out. But this isn't really all that clear. If a tree is healthy and it has enough reserves of carbon compounds, it can produce these resins and these monoterpenes and at really high concentrations. However, at low concentrations, the beetles are actually able to perceive that the tree is weak. And what that leads to is that the beetles being able to target that specific tree. So if a tree just isn't healthy, if it's stressed too much, if it doesn't have access to these carbon reserves that it needs, it won't be able to mount a successful attack. So these monoterpenes are very, very key in predicting how well these seedlings or these pine species will be able to protect itself against future attacks. So these three treatments really formed our three main research questions when looking at this. So considering that drought treatment or the water treatment, do seedlings mount monoterpene defenses under drought? So it is well known that adult seedlings are able to produce these, but that's also has been associated with bark beetles attacking primarily adult or mature species. Seedlings aren't really ever the target of beetle attacks because there's really just not much material there for them. So is this ability for adult pinyon pines to produce monoterpene defenses also seen at the seedling stage? Is it just a species throughout its entire life? Is it able to do this? Secondly, with the methyl jasmonate, are the trees that we spray with methyl jasmonate directly able to provoke some sort of defensive response? Is there evidence of defensive signaling? Could a tree next to another one that is being attacked perceive that that tree is being attacked and produce its own defenses? And then thirdly, with that pheromone, similar to the methyl jasmonate, can a tree directly detect the pheromone of a beetle and produce a defensive attack, or does it really need that physical damage to happen in order to produce a defensive response? So right now I'm going to go over some of the results that we have so far. We haven't analyzed all of the data so far from the VOC analysis, but we have a good basis to kind of see what's going on within these species. So just to show that our drought treatment was effective, we measured the water potential of our species. Now those letters down below indicate the treatments with water, MJ methyl jasmonate, pH being pheromone, DR drought. Those 4 treatments on the right were the droughted ones, and the 4 treatments on the left were the well-watered ones. And what you can see here is that the droughted trees had a much, much more negative water potential. What that means is the xylem column, which transports water throughout the tree, was under a lot more stress. It had a lot more pressure on it because the stomata were closed. all the water had pretty much left the tree. So the tension, there was a lot of tension in that column. So this is showing that that drought treatment was actually pretty effective. Now, when you juxtapose that with this current plot, which is showing just the total VOC concentrations, you can see that those droughted seedlings were producing on average a lot more in terms of compound concentration than those watered seedlings. So that's a very clear trend that we saw mainly along the droughted versus watered rather than with the other treatments. Now, when we do these analyses where we get these concentrations, we get more than 30 different types of compounds and concentrations which if you look at an Excel sheet are just a bunch of numbers and a bunch of names of a bunch of monoterpenes and sesquiterpenes and random compounds, which really don't make much sense. So you really need to use some sort of predictive model which can look at those concentrations and determine what concentrations of which compounds determine which treatment it went into. And that's exactly what random forest is doing. So on the left, we have a plot showing the relative importance. You can interpret that x-axis with the greater the number on the right being the more important that compound is in determining which treatment that tree belongs to. And what we did is we took those top 10 and just plotted them here on the right. And in that same trend that we saw in that previous slide kind of really shows itself here. So with the top row being watered seedlings and the bottom row being droughted seedlings, you can see that the droughted seedlings really are just producing a lot more of these monoterpenes, of these defensive compounds, than the water seedlings. And this can be interpreted with the droughted ones being the ones that typically face the stress from these predators. Now, that was just for the overall treatment, but we were also curious about each individual treatment and what determines the water versus drought, what determines the pheromone versus non-pheromone, and then similar to the methyl jasmonate. So these are the same types of plots. I caution you to look at those x-axes because that's kind of what shows you how important this is. With that water versus drought, there's a lot of compounds which have a relatively high number on the importance scale. And that's showing us that a lot of these compounds are determined by whether the tree receives water or not. Negative alpha, the needle alpha pinene concentration is a very important determinant of whether that is produced under droughted conditions. When you look over at that pheromone one, you can see while there are some on that right column, or on the right end of the scale, that the number really isn't that high. There aren't many of these compounds which are really jumping out at us and saying this compound is really strongly associated with a pheromone treatment or not. But when you look at methyl jasmonate, that's where it kind of steals the show, right? Methyl jasmonate is where we're trying to look at that signaling, defensive signaling within plants. And we see that on that top bar, that linalool, the compound, is extremely important, especially in comparison to all of these others. It's almost the only determining factor as to whether that tree received methyl jasmonate or not, as that concentration of linalool. So that kind of is our chemical spotlight for this project right now. This is just a bar plot of showing the linalool concentrations across the treatments. And what you can get from this is that when water is present and methyl jasmonate is present, linalool is produced. And there's that one dot on the drought and pheromone, but that's insignificant. It's really just under these conditions where this is being produced. Now, that is telling us that these trees are not stressed, right? They have plentiful water, and they're able to perceive this treatment which we impose on them, the physical spray of methyl jasmonate, and produce linalool. Now, linalool in other plants, such as rice, I believe, I read a paper about, is associated with plant pest defense. Now, like I said, what this means is that these plants can actually detect a signal from another plant. Theoretically, if the trees are close enough and if those methyl jasmonate concentrations are high enough in the air, a pinyon pine tree next to another one could detect that the neighbor one was under attack and be able to prepare its own defenses preemptively in order to better defend itself against these bark beetles. So this is what we have so far, but we definitely have more angles that we're going to be looking at in the future. So this is the going forward section. Now, non-structural carbohydrates are play a very important role in plant physiology. When I was talking about those carbon reserves, this is what I was talking about. So when a plant produces sugars through photosynthesis and it has a surplus for what it needs at that immediate time, it devotes some of that to storage. Now, those NSCs, which are storage, can be then used for growth, metabolism, repair, reproduction, or even defense response. So what we're curious about is that with this drought treatment that we had producing generally higher concentrations of these VOCs, are those coming from NSCs? Are the non-structural carbohydrates what's fueling that? Similarly, all of the data that we have so far is based on needle concentration analysis, but we also have woody tissue or stems, right? The area where the bark beetles actually bore into. So we still have to analyze that data to see if those compounds that are produced in the stem differ from that of the needle. And then thirdly, linalool, which is sort of, like I said, the star of the show. How important of it, what role could that actually play in the defense of pinyon pine to these bark beetles? All right, so that is everything that I had. Thank you for listening. I welcome any questions that you have, and you can definitely email me as well if you think of any later.
That's so cool. Okay, yes, I definitely have some questions for you.
Yeah. I can go back to that. Yeah.
Yeah, that would be great.
Yeah. Go for it if you can see it.
Okay. So my first question, I think, if you pull up, you had one where you had like the 4 non-drought and the 4 drought, and you had like the VOC concentrations.
Yep.
And
is this the correct plot?
I think so. It's still waiting for a second to show your screen.
Okay.
So, but so you're saying that if they have water, the linalool concentration is really high.
Yeah, let's go back to that one.
All the VOCs are pretty low with the water ones. Do you know why that is, or like?
So, do you see my screen right now with the chemical spotlight linalool?
I don't, but I'm not sure why.
Let me see if I can fix that. Oh, hold on. That was my fault. My bad.
No worries.
Here you are. Okay. Is this the correct plot that you're talking about?
I was looking at 2 slides above that, I think. One more.
Or this one here.
This one. Yeah.
Okay. So right, overall, the droughted trees are the ones that produce really, really high concentrations. But it's really just a question about relativity, like how big are the concentrations, and that's better described by this plot. So these are like the 10 most important concentrations, or the compounds, and with that purple section of those pie charts, you see that it's alpha pinene. That's a really, really large number of the total concentration, right? Of those top 10 compounds, especially in the drought ones, it's taking up almost 3/4 of the total concentration is attributed to alpha pinene. Whereas linalool, which is just a teal sliver, even though it is being produced in higher concentrations in that water methyl jasmonate and water pheromone methyl jasmonate treatments, it just really doesn't compare in total concentrations to the other ones. So that's probably where that's coming from.
But the relative concentration of linalool is higher when there's water, is what you're saying? Or
yes, but if you're focusing just on linalool, and you negate all the other ones, right? So each of these compounds have a different function. And they need different concentrations too, for whatever function they have. So when you're looking just at linalool, that's when you can kind of spotlight that it's being really produced in high concentrations relatively to the other treatments, which don't really produce any of it, under this water methyl jasmonate and water pheromone methyl jasmonate treatments.
So that might be the one that like plays the biggest role, or you suspect it plays the biggest role in like preventing the pine beetle attack.
It leads us to think that it has a role in just defensive signaling, right? Looking at this plot, that's when, if you're looking at the methyl jasmonate, the methyl jasmonate is just kind of a synonym right now for defensive signaling. Is there evidence of defensive signaling? We have a bunch of those compounds which kind of have more or less the same relatively low importance, and then we have linalool way out there as being the determining factor. So that makes us think that it is involved in some sort of defensive ability of pinyon pine seedlings, which correlates, like I said, to other species, not of pine though, which is super interesting. So this would be the first known, as far as I'm aware, the first known within species signaling of defensive chemicals for pine species.
That's so cool. So like theoretically, you could like, if you know that your pine beetle population is like worse than you want it to be for the ecosystem, you could maybe like spray the methyl, Yeah, like you could spray that into the air, and like the other ones might become stronger against the beetles.
Potentially, yeah. That's that's like a good ecological application, which I haven't really thought much about. But yeah, theoretically, if it's predicted to be a strong year, similarly, we use in some species we use these lures in like the Forest Service uses these lures to deter bark beetles. So like with white bark pine in Montana ecosystem, it's a threatened species under the Endangered Species Act. We really don't want them dying. So we actually put these lures which deter the beetles away from those trees. Similarly, that could theoretically be applied to defensive compounds in order to increase those defenses, almost like a vaccination for a tree.
Did you find, sorry, I'm a little unfamiliar with like the chemicals and things, so some of the plots take me a little extra long to figure out.
Yeah, no problem.
Did you find that they also respond with a defense response to pheromone?
So that's where we didn't really find anything. So with the importance one right here, there aren't really a lot. There are some that are theoretically important, but it wasn't really anything that stood out or is being determinate as well. So we're leaning towards it kind of not really being, at least in the seedling stage for our specific trial, not able to be detected. So going forward, I think we're going to focus mainly on the water and the methyl jasmonate treatment, or the drought and the methyl jasmonate variables, rather than also the pheromone variable.
Okay, that makes sense.
Yeah.
So like going forward, you talked about some of the experiments that you're going to do. Do you have any like hypotheses of like what you expect to see?
Yeah. So what's going to be really cool is the non-structural carbohydrates, because that's right now we have the trees dried and ground up, and we need to do this NSC analysis to see what are the relative concentrations of NSCs. Because it was a very short drought period. It was about a month that it took, and that is really short. So it's quite possible that the NSCs weren't able to be accessed because so in order to access these sugars and these growth NSC compounds, you need water. You need to be able to transport them. So when you have these drought conditions, it can get really hard, even if you have NSCs. If the tree dies of hydraulic failure, that means that it really won't be able to access these NSCs. Whereas if it's a sustained drought, then it can access them and help combat that drought. So it'll be interesting to see what the case was with our seedlings, whether these VOCs that were produced in higher concentrations for the droughted trees were coming from photosynthesis that was occurring at that time with those sugars, or if it was able to access some of those NSCs that it had stored.
So if it was able to access them, you would expect to see fewer NSCs?
Yeah, we would see a decrease in NSCs. Yeah, exactly.
Okay, that's very cool.
Yeah, definitely. It's exciting. Yeah, I'm looking forward to it. I have a bunch of data that I need to analyze still for it. But it's coming together, definitely. And it's really cool, especially for me, the defense signaling with linalool, because that would be, as far as I'm aware, the first known occurrence of that in pine trees, which would be pretty awesome to see.
Yeah, that would be so. That would be so cool. Do you expect then? So like the list that you had with the relative importance.
For like everyone, or for the individual treatments?
Yeah, for this. Yep. The fact that like linalool is so much higher, are you like kind of looking at an extensive list? So like linalool would be the one, or is there like other ones that you might also find later if you did different experiments?
That's where like, yeah, I mean, this is obviously just one trial with one defensive compound that is known in other species to work. And that's like, this is interesting because if you look at this next one, right, the big story when you just look at the total concentrations is that water determines it or drought determines it, right? That the relative concentrations of everything. But if you look at those important compounds on the water one or the drought treatment, linalool doesn't even show up in any of those. So it's interesting, or when it does, it's like down, it's barely in that top 10. Like it's maybe even looks like it's after that top 10. So it's relatively low on that one, but it plays a very important role on this methyl jasmonate. So like it would be hard to attribute anything to one chemical. Like they have different functions, right? They seem to respond to different sort of stimuli. So that's something to keep in mind.
Oh, okay. I think That makes a lot of sense. So like it's responding a lot defensively with water, but like specifically when it's defending when it gets the methyl jasmonate, then that's due to the lithanol. Okay, that's very cool.
Exactly.
Yeah. Sorry, it took me a second to kind of.
No. That's totally okay. I'm not a chemist at all.